Hello students, today our new chapter is Body Fluid and Circulation. It is the second chapter of Unit 5th, Human Physiology. Okay, now body fluids and circulation, we know very well we have two types of body fluids. One is blood and another one is lymph. Look here, in this chapter we are going to read about the blood and lymph, composition of blood, lymph and also the circulatory system of human. Okay, now here body fluid and circulation, you have learned that all living cells have to be provided with nutrients and other essential substances, and also in our body, due to the result of metabolic activities, we have the waste or harmful substances which are produced. They have to be removed continuously for healthy functioning in our body. Okay, therefore it is essential to have efficient mechanism for movement of these substances to the cell and from the cell, one cell to another cell, and different organisms. And now when we consider the different organisms, because we know very well we have unicellular organisms, multicellular organisms. In multicellular organisms we have different type of level of differentiation, that is body level of differentiation, cellular level of body organization, or second one is our tissue level of body organization, or organ level of body organization, or organ system level of our body organization. We have different type of organization, and different type of organization, that means they have different type of systems. Clear? Now it is given here simple simple organisms. Simple, that means we have porifers, aspirants and coelentrates. They are simplest. So aspirants, they have cellular level of organization, and coelentrates, they have tissue level of organization. Circulate water from surroundings through their body cavities. That means in case of aspirant we have canal system and aspirantocele cavity is there. In coelentrate we have coelentron cavity is there, which is called gastrointestinal cavity. So they facilitates this function of circulation through their body cavity. Another more complex organisms use special fluid within body to transport such materials. They have, that is, they have a different type of systems or organs which are responsible for transportation. And the blood in us, blood is the most commonly used fluid by most of the higher organisms. In animals, complex animals, we have tissue, that is blood. Blood is the most commonly used tissue for circulation or for transportation of different fluids. The most higher organisms include human for this. Okay, another body fluid is lymph, and which also helps in transport of certain substances. Maximum certain substances are transported through blood, but some substances they are transported through lymph. Okay, and in this chapter you will learn about the composition and properties of blood and lymph tissue fluids, and their mechanism of circulation is also explained herein. It is given in NCERT. Okay, now it is blood. Now here we have two tissues. First one is blood. First thing is, what is the, that means study of blood. The study of blood is called hematology. Hematology, that means study of blood, and its composition also, it is its property. Okay, another point is blood formation. Blood formation is called hemopoiesis. Responsible hormone or enzyme is hemopoietin. Clear? Now blood. What is the composition of blood? When we consider the blood, that means blood, it is made up of two components. First one is plasma, which is fluid, And second one is blood cells. In NCERT it is called formed elements. That means blood cells. Formed elements or blood cells. In formed elements we have RBCs, WBCs and platelets. Okay, and in case of plasma we have water and another substances, that may be protein or others, and other substances. Okay, when we consider the plasma, what is the portion of plasma? Plasma, 55% of blood is made up of plasma or blood fluid, and 45% it is made up of formed elements. Formed elements. Clear? Okay, now when we consider the blood plasma, as I told you, blood plasma, composition of blood plasma, it forms 55% of total blood, and it has two main components. One is H2O, that is water. Water, it is 90 to 92% of plasma, and remaining substances contains mostly proteins and other substances like organic or inorganic, organic and inorganic substances. Okay, when we consider this protein, it makes 6 to 8% of total protein, and the remaining 1% they are organic or inorganic substances, less than 1%. And protein, we have three types of proteins, mainly three types of protein which are found in our blood, and they are, first one, the maximum protein is albumin type of protein. Albumin protein. Albumin protein, it is 4% protein. Clear? Total 4%. And what is the function of albumin? It regulates osmoregulation of blood. It is responsible for the osmoregulation. So it maintains the BCOP means blood colloidal osmotic pressure. It maintains the blood colloidal osmotic pressure, and always it made it 28 to 32 mmHg. This one is osmotic pressure. Clear? Which helps in also excretion. So it means albumin is responsible for maintain the BCOP, that means blood colloidal osmotic pressure. Second protein is globin. Globin or globulin, we may sometimes say them globulin protein. It makes 2% of total blood. That means what is the ratio of albumin and globulin? For albumin and globulin, ratio is 2:1. Albumin and globulin or globin. Globulin or globin. Okay. Now, globulin protein, that is globulin or globin protein, it is total 2% in the blood. It is of three types. It is of three types. Mainly it is two types, but when we consider it is we consider it in three types. So first one is hemoglobin. Globin, that means first is one, that is copper, like copper globin protein. It synthesized in liver. This one is copper containing, first copper containing globulin, second one is another globulin which contains iron. Iron is Fe plus 2. Only Fe plus 2 is present in blood, not Fe plus 3. Okay. Globulin, it also synthesis, that is formed in synthesis in liver. Another third one is globulin or globulin. It is synthesized in, that which is produced in lymphoid organs or spleen also. That means it is related to immunity, and it is present in our antibodies. Clear? So sometimes we said them because it is related with immunity, immune system. So it is also called as immunoglobulin. And represented by Ig. Okay. So when we consider the immunoglobulin, we have also 5 types of immunoglobulins, and these 5 types are Ig. Ig means immunoglobulin. They are first IgA or IgD or IgE or IgG, or they may be IgM. Clear? Now, it is also represented by alpha immunoglobulin, D delta immunoglobulin, E sigma immunoglobulin, G gamma immunoglobulin, or M mu immunoglobulin. This is also, that means we use in the form of Latin language. Okay. IgA, IgD, IgE, IgG or IgM, they are antibodies, the different type of antibodies found in our bodies. And in IgA, we know it is found in colostrum, the first milk. IgA, IgD, E for allergy, M for myasthenia, that is muscular. So we have 5 types of immunoglobulin. Immunoglobulin is synthesized in our immune system, that means lymphoid organs. Other which is synthesis in or which is produced in our liver, so they forms hemoglobin. Hemo, that means it contains iron molecules. Okay. Now this, after globin protein, we have third, it is fibrinogen protein. Fibrinogen, it is soluble form in blood. It is a fibrin, that is, it is monomer. This one is monomer of fibrin protein. Okay, at the time of blood clotting, it forms polymer and forms fibrin. Then blood clot occurs. Another one is there, it is heparin. Heparin, it is also a protein, but it is very less in amount. That means it contains only 0.3%, and it also 0.3%. They both 0.3%. 2% is our globin protein, 4% is our albumin protein. So heparin, it is an anticoagulant agent. So it helps to prevent the coagulating of blood. So these are the proteins, different type of proteins which are found in our blood portion. Okay. Now another, we have organic and inorganic substances. So in case of organic or inorganic substance, we know very well blood, the main function of blood is to transport the nutrients, or also it transport the waste products. That means in blood it contains ions, sodium, chloride, potassium, bicarbonate. Also it has enzymes like carbonic anhydrase enzymes. So we write only enzymes, hormones, then substances which are always present in the form of transportation. They are glucose, clear, amino acids. Excretory products also gases. In case of gases, CO2 and O2. Clear. Excretory products, like urea, etc. So they are also present in our blood. So this is the total composition of our plasma, that is 55%, which is the 55% of our total blood. Here what is given in NCERT? Blood. Blood is a special connective tissue consisting of fluid matrix and formed elements. So fluid matrix, that means what? That means plasma, and formed elements, that means RBC, WBC and platelets. So two parts in blood. First one is plasma. Plasma is a straw colored pale yellow. Straw means pale yellow colored viscous fluid constituting near 55% of the total blood. 90 to 92% of plasma is water, as we already read here. Okay, and proteins contribute is 6 to 8% is total protein in it. Fibrinogen, globulin, albumin are the major proteins. These three proteins which I told you: fibrinogen 0.3%, globulins they are 2%, and albumins these are the maximum that means form of albumins, that means 4% are the major proteins. Okay, I will tell you albumins 4%, globulins that means 2%, and fibrinogen they are 0.3%. Another protein is heparin. So main three proteins are this: albumins, globulins and fibrinogen. Okay, in case of, as I discussed, so globulin is we have three types of globulin. One is copper containing globulin, another one is ferrous iron containing globulin, and another one is immunoglobulin. Okay, both first two, that means copper and iron, they are formed in our produced by liver. They are formed in our liver. And albumins they are formed or they are produced by our lymphoid organs or spleens. Now fibrinogen are needed for clotting. Third one is fibrinogen, and it is given here first. So fibrinogen are needed for clotting. I told you fibrinogen protein, it is in soluble form of blood because it is monomer. Clear? When it helps in coagulation, that means when it helps in coagulation, these monomers of this fibrinogen protein are polymerized and forms fiber-like substances. And these fiber forms a network and clotting obtained. Okay, globulins primarily are involved in defense mechanism, as I already told you. Globulin primarily are involved in defense mechanism. Clear? The primary function of globulin is defense mechanism, that is immunity. So they are found in our antibodies in the form of immunoglobulin. Clear? And I told you immunoglobulin. How many types of immunoglobulin are there? There are 5 types of immunoglobulin: IgA, IgD, IgE, IgG, IgM. Okay. Different of the body. And albumin. Now another one is albumin. Albumin, which is 4%. Okay. It helps in osmotic balance, and here it is called BCOP. BCOP means blood colloidal osmotic pressure. That is, it maintains it or it constant in the ratio. Okay. Globulin is protein. It is, what is the primary function of globulin? it is immunity. Clear? That means it is found in our body in the form of antibodies. And we have 5 types of antibodies: IgA, IgD, IgE, IgM and IgG. Clear? Second, globulin. It is in the form of hemoglobin, that is transportation of gases, oxygen or carbon dioxide. Okay, now albumin, which is maximum in our body, that is 4% is albumin. Albumin, it helps in osmotic balance, that means it maintains the peak of blood colloidal osmotic pressure between 28 to 30 mmHg. Plasma also contains, now we have some another minerals, that is organic or inorganic substances. So plasma also contains small amount of minerals like sodium, calcium, magnesium, bicarbonates or chlorides. Now glucose, amino acids, lipids, organic substances are also present in our blood, also present in the plasma, and they are always in transit in the bodies. In transit, that is they are moving particles or responsible for transportation. Factors for coagulation or clotting of blood are also present in the plasma. We know very well 13 factors are involved in blood clotting, which we read further more. Okay, so these factors are also present in the blood plasma, but they are present in inactive form. Okay, plasma without clotting factor. Plasma without clotting factor is called serum. Plasma without clotting factor is called serum. That is what? When we minus plasma, that means plasma minus what? Clotting factors. So we obtained serum. And what is plasma? That means blood minus formed elements. Then we obtained plasma. Clear? So plasma, that means if we separate the formed elements from blood, remaining part is called plasma. And when we separate clotting factors, that means this fibrin protein or other, so the remaining part is called serum. Now we have formed elements. Clear? Second one is our formed elements. So formed elements, it constitutes 45% of total blood. Plasma 55, formed elements 45% of blood. Look here, erythrocytes, leukocytes and platelets are collectively called as formed elements, and they constitute nearly 45% of the blood. Clear? Erythrocytes, now first one is erythrocytes, that is called RBC. First one is a formed elements, they contains RBCs, second WBCs, and third one is our platelets. So first one is our RBC, RBCs, they are called as erythrocytes. Erythrocytes. Erythrocytes, red blood cells. They are most, first was this, they are most abundant. Cells in our body, in our blood. Clear? In case of human males contains 5.5 6.5 to 5.5 million blood RBCs in per cubic millimeter. Okay, in per cubic millimeter, that means mm square. So cube. Clear? That means what? Erythrocytes are most abundant. They are 5.5 million in per cubic millimeter in case of male, but in case of female, they are some less. They may be 5.5 to 4.5 million per cubic millimeter. When we consider the fetus, that means in embryonic fetus, they have more, that means 6.8 million per cubic millimeter. Most evident, they are very small. Okay, now we have some other properties of WBC, like what is the structure of RBC. So mammalian RBC is biconcave structure. Biconcave structure. Biconcave means this type of structure. This type of structure. Why this type of structure? So it increases. First thing, it increases biconcave structure. It increases surface to increase surface area. Why it is biconcave? To increase by surface area, and because they are enucleated, that means nucleus is absent in mammalian. Two exceptions are there, and they are, first one is camel, another one is llama. Okay, if nucleus is there, they are not biconcave, they becomes convex. That means if nucleus is there, so they becomes convex because there is nucleus. Clear? So in case of camel and llama, the RBC is biconvex, and nucleus is also present. But in case of other mammals, they are exception. Camel and llama, they both are exception. Okay, but in other mammals, only mammals. Clear? RBC is biconcave structure. Biconcave structure. No RBC is there. No membranous cellular organelles are there. Newborn RBC have all the cell organelles, but when it get mature, all the cell organelles get disappeared. Only hemoglobin is there. Clear? Because it has no nucleus, it has no other organelles. That means it cannot reproduce or multiply. Okay, when we consider the membrane of RBCs, that is plasma membrane of RBCs, so they are called as Dickan membrane. Why they are called Dickan membrane? Because they are permeable to chloride and bicarbonate, but impermeable to like sodium or potassium. So they are called as Dickan membrane, Dickan type of membrane. Because RBC have no cell organelles, no endoplasmic reticulum is there. So they have cytoskeleton made up of that is not a big bus but fatty acids cholesterol, that means it is spongin. A spongin skeleton is there. Spongin skeleton is present. Look, a spongy skeleton, because it is a spongin skeleton, so it can increase or it can decrease its size. So the size of RBC, when we consider the size of RBC, it is 7.5 micron. Clear? Okay. When we consider the blood capillary size, so size of blood capillary is 5 micron. Then how can it pass through blood capillary? Due to its a spongy nature, it can pass with the small diameter of blood capillaries. Clear? So size is greater than blood capillaries diameter, but it can pass through this blood capillary because due to its spongy nature. Okay, now we know now they are non-nucleated. We know what I am writing here: non-nucleated, no membranous cell organelles are present. That means all are absent. All these are absent. So this is about erythrocytes. What is given in NCERT? Now we are going to look in the NCERT. What is given here? Look, formed elements, erythrocytes. Erythrocytes, leukocytes and platelets are collectively called as erythrocytes, leukocytes and platelets. They are collectively called as formed elements, and they constitute about 45% of total blood. First thing is there. Second, erythrocytes or RBCs are the most abundant of the cell in the blood, and a healthy adult man on average 5 million to 5.5 million of RBC per cubic millimeter they are found in blood. Okay, we are right here now. In the previous, so I told you in male it is 5.5, in female it is 4.5, in fetus it is maximum, it is 6.8 million per cubic millimeter. Okay, RBCs are formed in red bone marrow. Okay, this one is important. Where RBCs are formed? In red bone marrow. And where is the red bone marrow? Red bone marrow is found in our spongy bones. A spongy bones, that means these are epiphyses of our bones. Clear? So they are found not in the shaft. This is not. They are found in trabeculae, that is the small vacuolized structure. Small space are there, and these spaces are called trabeculae. This bone marrow is filled in the trabeculae, red bone marrow, and it is responsible for the formation of RBC. Okay, RBCs are formed in red bone marrow, in the adults. Okay, in adults it is red bone marrow. RBCs are first RBC is formed in fetus by yolk cell. Okay, RBC are devoid of nucleus, that means nucleus is absent in most of the mammalian, and are biconcave. So they are biconcave in shape. Why here said most of the mammals? Because we have two exceptions: camel and llama. They are also mammal, but they have biconvex erythrocytes. Okay, and they have also nucleus, because they have nucleus. Their blood cells have nucleus, so their shape is biconvex. Okay, and all the other they are biconcave in shape. They have a red colored iron containing complex protein which is called hemoglobin. Okay, look here, hemoglobin about. Now another point is hemoglobin. Hemoglobin is made up of two constituents. First one is heme, and another one is globin, protein. Heme, it is iron or porphyrin. Clear? And globin, it is globin proteins made up of two polypeptide chains. Two polypeptide chain. The first one is our iron or porphyrin ring, clear, which is called heme, and another one is globin. Now globin, this one we have three types of hemoglobin. Like first one is hemoglobin A, hemoglobin A2, and fetal hemoglobin HbA1. Second one is our HbA2, and third one is our HbF. HbA1, in all these three, we have four types of polypeptide chain, and these four types of polypeptide chains are alpha, beta, gamma, and delta. Clear? Four types of polypeptide chains. Now, HbA1, it is found in adult. It is made up of two alpha polypeptide plus two beta polypeptide. A2 made up of two alpha plus two gamma polypeptide. And in case of fetus, it is two alpha plus two delta polypeptides. So we have, that means, four chains made up of two different type of chains. Clear? Now it is clear. HbA1, HbA2 or HbF. In these three, alpha chain is same, identical, and the second beta in case of HbA1, in case of HbA2 it is gamma, and in case of HbF, F means fetus. This one is fetus. For fetus. It has more capacity to carry oxygen. Okay. Now another point. Some specific conditions of RBCs. RBC can change its size. When its size is changed, when change in size, so this one is called N-iso. Iso means identical to N. Because it is cell, N-isocytosis. Change in size, it is called N-isocytosis. When change in size, that means what? It can increase its size or it can decrease in size. When it increase in size, it is called macrocytosis. When it decrease in size, it is called microcytosis. Simple. Macro means large and micro means small. When it increase its size, so due to the deficiency of vitamin B12, due to the deficiency of vitamin B12, clear? B12, it increases its size. Size is increased. Size is increased, so it is called as macrocytosis. Now, in case of this, heme is equal. Heme is normal. That means, in this case, heme is normal. Heme, it is normal. But size is clear. Second thing, and when it increases size, it is destroyed by our spleen, because it becomes abnormal. Okay. When due to the, another thing is, when this is the increase in size, that is macrocytosis. And what is the example of decreasing size? So another one is due to the deficiency of iron, Fe plus 2. Clear? Iron size is decreased. Size is decreased. And when size is decreased, it is called as microcytosis. Microcytosis. In this case, because Fe is decreased, so heme is the part of that is hemoglobin. That means heme is less. Heme is less. Because its size is also decreased, it also becomes abnormal. So it is also destroyed by our spleen. Okay, so this is called anisocytosis. That means increase in size, macrocytosis, or decrease in size, that is called microcytosis. Clear? Okay, now once again here. Size change, it is called anisocytosis. When shape change, shape means it is biconcave or now it becomes biconvex. That is called shape. Size is another thing and shape is another thing. When shape is changed, so it is called poikilocytosis. You very well know, familiar with the poikilo word, poikilo term. Poikilo means which can be changed. Poikilothermal elements like. Okay. So poikilocytosis, that means changes shape changes, and it occurs when we put RBCs into hypo or hypertonic solution. When we placed this RBC in hypertonic, this one is hypo. In hypertonic, what happens? This cell shrinks. And when we placed it in hypotonic, then it swelled up and ultimately burst. Clear? So this is called poikilocytosis. Now another terms according to their shape or size, that means when another term is shape, size, or its number. So when the RBCs are decreased in number, so this abnormality is called anemia. Decrease in number, it is called anemia. Anemia is also of 3 types. It is also of 3 types. Clear? The first one is our macro, when vitamin B12 deficiency is there. Its size is increased, but content of heme is normal. Because heme content is normal, so we set them size increase. So I write here macro, normo. Now chromo is Fe, chromo anemia. Clear? Macro, normo, chromo anemia due to the deficiency of vitamin B12. Second one is that means size is increased, but chrome is that is heme is normal. Second one is micro. Now it is not normal, it is hypo means less, chromo anemia. Clear? Micro hypochromo anemia, that is what it is due to the deficiency of iron, Fe2. Clear? So 2 types of anemia, and another one is which is normal in structure, normal in size, but due to the blood loss, due to an injury, more blood loss is there, and this is called normocytic anemia. Okay, that means normocytic is called normocytic. Cytic means cell, cell is normal. Second one is normo, this is anemia, but anemia is there due to the maximum water loss in an accident or in an, everything is normal due to the excessive blood loss. Excessive blood loss. We have three types of anemia. Clear? Now it is clear. Okay. Now we once more when we look into the NCERT. Here it is. RBCs are formed in the red bone marrow. I told you where red bone marrow are there, in a spongy bones, and as spongy bones they have trabeculae, and these trabecules are filled up with red bone marrow. There RBC are formed. Second one is RBC are devoid of nucleus in most of the mammals except two, camels and llama, and are biconcave in shape. Clear? But in camel and llama they are biconvex because they are nucleated. They have a red colored iron containing complex protein which is called hemoglobin, and which we already read in this part. Clear? That means we have heme plus globin. Globin is also, we have three types of hemoglobin, HA, HB, that means hemoglobin A1, hemoglobin A2 and hemoglobin F. Clear? And globin protein, it is made up of 4 chains, that means 2 different type of polypeptide chains, 2 alpha plus 2 beta, or 2 alpha plus 2 gamma, or 2 alpha or 2 delta. Clear? Hemoglobin has the color and the name of the cell because it contains hemoglobin. So red in color due to the presence of heme, that is iron, it is red in color. Clear? And due to the presence of copper, it becomes bluish in color. Okay. A healthy individuals have 12 to 16 gram of hemoglobin in each 100 grams. Each 100 gram of blood contains 12 to 16 gram of hemoglobin. Now consider 12 to 16 gram of hemoglobin is present in 100 ml of blood, and we know very well each gram of hemoglobin is responsible for the transportation of how much oxygen? 1.33. Clear? Now look here, 12.16 gram of hemoglobin in each 100 ml of blood, and 1 gram of hemoglobin, clear, transport 1.33 gram of oxygen. That means 12 to 16 gram of hemoglobin transported 20 ml of oxygen. 20 ml of oxygen, clear? 20 ml of oxygen, that means 100 ml of blood transported 20 ml of oxygen. Clear? So these molecules play a significant role in transportation of respiratory gases, either it is CO2 or it is oxygen. We know very well our 97% of oxygen is transported through this hemoglobin, and at least 25%, 20-25% carbon dioxide is also transported through hemoglobin in the form of carboaminohemoglobin, and in case of oxygen it is in the form of oxyhemoglobin. These molecules play a significant role in transportation of respiratory gases, that means CO2 and O2. Now RBC have an average lifespan of 120 days. It is average lifespan of 120 to 127 days, but in NCERT it is even 120, so you consider the 120 days. Clear? And after they are destroyed in spleen, they are destroyed in spleen. So spleen is also called as graveyard of the RBC. Spleen is also called as graveyard of RBC. Spleen is called graveyard of RBC. Spleen is also called as blood bowl. Why? Because it also stores the RBCs, extra RBCs. It can store RBC, it can destroy the abnormal RBCs. Clear? As we read here, in case of macronormochromatic anemia, what is there? They are, because their size is abnormal, they are large in size, so spleen separate them and destroy them. In case of microhypochromatic anemia, when deficiency of iron is there, their size is small, they also captured and destroyed by spleen. Clear? So this is the spleen's function. Okay, the spleen is also called as graveyard of RBCs or blood bowl of RBCs. Now another one is our leukocytes. Leukocytes means WBC. Leukocytes are also known as white blood cells because leuko means colorless, as they are colorless. Why called white blood cells? Because they are colorless. White blood cells, as they are colorless due to the lack of hemoglobin. They have no hemoglobin. That means they have no globin proteins. They are, one thing, nucleated. They are nucleated and they are relatively lesser in number also. Okay, RBC, what is the count of RBC? It is 5 to 5.5 million per cubic millimeter. But what is the number? It is lesser number, which average 6,000 to 8,000 cubic millimeter of blood leukocytes. Clear? And they are generally short-lived. What is the lifespan of RBC? It is 120 days. What is the life of WBCs? It is different. That means some leukocytes are for 6 hours and some are for 6 to 7 days. Variable. We have two main categories of WBC, two main categories according to their cytoplasm. One is called granulocytes, granular cytoplasm, and another one is called granulocytes. That means cytoplasm is clear, no granules are there. Now here, in case of granulocytes, we have three types of which WBCs, that are neutrophils, eosinophils, and basophils. Neutro means neutral, that means what? They can be stained either in acidic or in basic medium. Second, eosinophils, they are also called as acidophils because they can stain only in acids, acidic stains like eosine, so they are called as eosinophils. And basophils, basophils means that they can stain only in basic stains or alkaline stains like methylene blue. Clear? So according to their staining, different stains, they are different staining bodies. Second, different type of nucleus size also, they are classified in three divisions, that means neutrophil, eosinophil, and basophils. Clear? Most evident is neutrophil. It is given in after them, it is 60 to 65%. Eosinophils is only 2 to 6%, and basophils, they are minimum in number, that means 0.5 to 1% of total leukocyte TLC. They are the different types of granulocytes. So granulocytes, three types of granulocytes are there: neutrophils, eosinophils, and basophils. Sometimes we said neutrophils, acidophils, and basophils. Okay. While lymphocytes, we have two another: lymphocytes and monocytes. Lymphocytes and monocytes, they are granulocytes. That means what? We have 5 types of WBC, 3 types granulocyte, and 2 types agranulocytes. 3 types granulocytes: acidophils, basophils, and neutrophils. 2 agranulocytes are monocytes and lymphocytes. Neutrophils are most evident. Look here. Neutrophils are most evident cells. That means they contain 60 to 65% of total WBCs. And basophils are least in number. That means they constitute 0.5 to 1% of total WBC. Clear? Second one is neutrophils and most monocytes. Neutrophils and monocytes. So neutrophils, they are 60%, but monocytes, they are only 6 to 8% in our body. That means in total TLC, are phagocytic in nature, That means they are also called like scavenger. Okay, because neutrophils, they are smaller in size, so they are called as micro policemen of our blood circulation. Monocytes, because they are larger in number, macrophages, so they are also called as macro policemen of our body circulation, and are phagocytic in nature. So they will destroy the foreign organisms entering in our body. Clear? That is biological immunity or biological level of defense. They create biological level of defense. Another one is basophil, which is least in number, secretes some chemical that means histamine, serotonin and heparin. Histamine, serotonin and heparin. They are also secreted by mast cells. So these cells are basophil cells are acts as mast cell. Clear? Mast cell we will study it in tissue. So histamine, serotonin, heparin, that is one, it acts as vasoconstrictor, another acts as vasodilator, and heparin we know it is anticoagulant in blood. Okay, and are involved in inflammatory reactions. Clear? Now eosinophils, it is responsible for allergen also. Clear? So it constitutes 2 to 3%, resist infection and are responsible for allergic reactions, associated with allergic reactions. Clear? Lymphocytes, second largest that is in case of number. Clear? It constitutes 20-25% and of two major types. It is B or T, lymphocyte B or lymphocyte T. B for bone marrow and T for thymus gland. Trained lymphocytes which are trained into the thymus gland in the presence of thymosin hormone. Clear? They are called lymphocyte T. So lymphocyte T is responsible for cellular mediated immunity, CMI, and lymphocyte B, which forms antibodies. It creates in our body AMI type of defense, that means antibody mediated immunity. So two major types B and T forms. Both B and T lymphocytes responsible for immune responses of our body. Acute. So they are responsible for acute defense line of our body. Now another is platelets, also called as thrombocytes. Now we have B and T forms, and they 25% responsible for immune response of our body. I very well told you B is responsible for the formation of antibodies and T is responsible for cellular mediated immunity. The third one is our blood platelets. The platelets are called as thrombocytes because they contain thromboplastins. Thrombostatin protein is present in platelets. Are cell fragments. They are cell fragments produced from megakaryocytes. Mega means large, karyo means nucleus cells. Megakaryocytes, special cells, they are found in our bone marrow. Special cells which are found in our bone marrow. Clear? So they are the cell fragments produced in the megakaryocytes of our bone marrow. Normally blood contains 1,50,000 to 3,50,000 platelets per cubic millimeter. It is the range, normal range. Clear? Okay. When these platelets decreased and come up to 40,000 per cubic millimeter, so this one is very critical condition, and when platelets become low, that means it become lower than 40,000. So this type of disease, this disease called purpura disease. At this time, red spots or rashes are seen in our skin. Okay, purpura disease due to the absence of or due to the decrease in platelets. Platelet can release various of substances, most of which are involved in coagulation or clotting. That means it is responsible to those because it releases thromboplastin. Clear? So it is also, so this is called thrombocytes. Its main function to blood clotting. First, a reduction in their number can lead to clotting disorders, clear, which will lead to excessive loss of blood from the body. So they are also responsible for blood clot. Blood clot occurs when we have some wounds, when our vessels are cut down. When these platelets come in the contact of air, they burst and release thromboplastin. Okay. So these are that means platelets, thromboplastin. Our cells are also release thromboplastin. Sometimes what happens is in our body in vessels, clot is there. When these clots are there, they are called thrombus. So we have two types of clots. Thrombus, when they are formed in our body, so they are responsible that they can cause block our vessels. If they block our cranial vessels, so it may leads to paralysis. If they clot our coronary arteries, so that means it may cause for heart attack also, which is fatal. Okay. How many minutes? 1 hour complete. Now WBCs. Leukocyte, commonly called as leukocyte. Clear? Leukocytes. So they are of two types. First one is granulocyte. Granulocytoplasm. So these granulocytes are three types. We need not to go very deep. Only according to SCRT we have to read three types. So these three types. The first one is our eosinophils or eosinophils. Eosinophils, they are also called as acidophils. Acidophilus or acidophils, they are responsible for resist infections and allergy reactions. Eosinophils or acidophils, they are actually bilobed nucleus. They have a bilobed nucleus. They have bilobed nucleus. Clear? Second, another thing is they can stain in acidic medium. That means we use eosine. So we said them eosinophilus. Clear? When that means they are hypersensitivity of acidophils is called this one, eosinophilia. That is a type of allergy. Clear? Eosinophils. Okay, what is the constitutes of it? Is total 2 to 3% of total leukocyte count. 2 to 3%. Eosinophils. Now another one is our basophils. Second is basophils. So basophils stain in basic medium. Basic medium, that is in methylene blue. Methylene blue. So they are called basophils. So they secretes chemicals like we have serotonin, histamine, or heparin. And by this action of these chemicals, they are inflammatory reactions. Clear? So one is vasodilator, that means vasodilator, vasoconstrictor, and this one heparin, it is anticoagulant. And now another is what is the composition of it? Constitutes very less, that means they are minimum in number 0.5 to 1. 0.5 to 1% of total leukocyte count. Minimum in number. Structured, what is the difference between the nucleus? So they are three-lobed or S-shaped nucleus. Shaped nucleus. S-shaped nucleus. Clear? Another one is our neutrophils. Third one is neutrophils. Neutrophils, that means they can stain acidic as well as basic stains. So they are called neutral. Maximum number in blood, that means it contains 60 to 65% of total leukocyte count. Clear? So they are phagocytic in nature. Phagocytic in nature, that means they can decompose or they can directly attack to pathogens and also engulf the debris of blood. So they are called as macro policemen of blood. They are 5 lobe structures: 1, 2, 3, 4, and 5 lobe nucleus. 5 lobed nucleus. Because they are, in the case of granulocytes, we find different type of nucleus, different lobes of nucleus. So we also call them, all the granulocytes, they are polymorphonuclear. Polymorphonuclear WBCs. Poly means many, morpho means morphology, that means structure of nucleus. WBC. Now we have another granulocytes. Granulocytes. That means clean and clear cytoplasm. So we have 2 types. Granulocytes are 2 types. 2 types. One is our monocytes. MONO, monocytes. They comprise 6 to 8 percent of total leukocyte cells. Okay, and monocyte, their structure, now they have only single lobe. That is, all the granulocytes, they have single nucleus, but nucleus is some kidney shaped nucleus. They have a kidney shaped nucleus. They are large in size, so they are also called as macrophages. Macrophages, or sometimes they are called as chief cells. Chief cells. Clear? They are also called as scavengers. Scavengers, because they eat the debris of our dead or debris or dead cells in our blood circulation. So they are also called as sweeper of our blood cells, macrophages. Clear? They are micro policemen of blood. And last one is our second, lymphocytes. Last one is second, lymphocytes. We have two types of lymphocytes, and it constitutes 20 to 25% of TLC, total lymphocyte count. Two types. First one is our B lymphocyte, and second one is our T lymphocyte. B lymphocyte, B for bone marrow. They are responsible for AMI, that means antibody, because they produce antibody mediated immunity. Another one is our T lymphocytes. They create CMI, that means what? Cellular mediated immunity. B lymphocyte is responsible for producing antibodies. They produce antibodies. But T lymphocytes, they directly attack. They are called killer cells. They have specific functions. So they may be killer cells, suppressor cells, helper cells, memory cells. Killer cells, they attack. They are soldiers. That means they attack to the pathogen, directly attack to the pathogen. Suppressor cell, they suppress the soldier cells. Because they are very aggressive in number. Then helper cell, they stimulates B lymphocytes to secrete antibodies. Clear? And memory cells, that means they are these killer and suppressor cells, that means they can memorize the pathogens antigen. Pathogens, that means it is called antigen. It is present in pathogen. So these are the different type of our WBCs. Clear? Another one is our platelets, which is called thrombocytes. So it is all about our blood and its composition.